So this is something that Sakana AI released recently. This is called Petri dish NCA. This is neurocellular automata work that they've been working on. So neurocellular automata is a relatively new type of cellular automata. If you're familiar, there was a paper in Distill Journal several years ago. Michael Levin was on it, and a couple other people, and they developed this method for looking at, or basically having a cellular automata, which is a pretty old approach to computing. It dates back to the 50s and 60s, and they were using cellular. They've used it in physics even, where there are these grids that have cells where you have neighborhoods, so you have a cell that has neighbors, and those neighborhoods govern the sort of state of a focal cell in that neighborhood, and then the neighborhoods are arrayed in parallel. So you can basically, the state of each cell is governed by its neighbors, and things can propagate across the topology of the automata or the grid. So each automaton, which is a cell, can affect other neighboring cells, and those effects can propagate across the space. And so you end up with these different types of structures that emerge. So things like gliders or other types of meta organizational structures. And so what NCA is basically for each cell, you have some neural agent or some neural network inside of that cell. So every cell has a neural network. It has some sort of neural controller, and it's basically, in this case, this is a bunch of convolutional neural networks that are arrayed in parallel, and they're obeying the rules of the cellular automata, and they're doing all this stuff. So if you're familiar with the work of Bert Chan, who works out of Japan, he was another early person who was working on neural cellular automata. And this is something that Sakana AI has developed. It's a demo of Petri dish NCA. So this is just a demo, and we're going to walk through this demo. So with all that in mind that I told you about how this works, I want you to take a look at this simulation. So we're compiling this in a GPU, and here is our NCA. So we're trying to minimize our loss as we run this simulation. Remember, each cell, and you can see these squares, if you have a single square, for example, that's a cell. So you can see this, here's a cell, and that from that green in a single cell sometimes propagates out to a block of cells that are of all the same color. So basically, you're trying to evolve shapes, or you're trying to run the simulation so that you can get these metastructures like shapes or gliders which move across the space. You're also trying to minimize the loss, which means you're trying to sort of refine these structures at the single cell level. And then, that's something that you can train. You can train these models to do things like replicate morphogenesis, which was what they did in the Distill Journal article on this. So this is a demo where they're just, we're running the simulation. We're not really minimizing our loss very much here, but it's running. So it's basically running through the different steps. I can look at what's happening with this step by step in here. So actually, we have two measurements here, because this is a it's a neural network model where we're trying to minimize our loss, but it's also sort of an evolutionary model where we're trying to increase our diversity. And so if we increase our diversity, we're replicating life more than we're replicating sort of an optimized solution to something. So we have our diversity and our loss for each step. And so we can then play with the, so let's pause it. We can then play with the different parameters, so we can increase the grid size quite a bit. Let's see if we can do this. Okay, so we're going to start simulating this much larger space. It's going to take longer to start up. But basically, it's trying to reproduce patterns that you might see in living systems. If you're familiar with, of course, Stephen Wolfram's work on A New Kind of Science, his proposal is that you can take all of nature and reduce it to cellular automata. That you can basically explain life and nature itself through cellular automata. And in a normal cellular automata, you have these rules that each cell follows. So each cell is an agent, and it doesn't have a neural network inside. It just has a rule that it follows. And so usually, if it's like there's like rule 10 and rule 30 and rule 110, and all those different rules are just like logical conditions or logic gates that basically have to be true in order for something to be on or off. And so when it's on, it's lit up. When it's off, it's dark. And so if those rules are set to condition whether it's on or off, you can produce these patterns that look like things in nature. So I think rule 30 and rule 110 have been used to sort of explain patterns on seashells. So if you've ever looked at a seashell, it kind of looks like the output of a of a cellular automata, and so the idea is that there's this correspondence between what the cellular automata is doing and what nature is doing with a patterning in a seashell. There are other types of patterns that are very interesting looking. You can generate all sorts of patterns like snow, kind of like things that look like snowflakes or things that look like other types of patterns in nature. Now, what Wolfram was arguing is that there are these rules that exist in nature that correspond to rules in a cellular automata, and that's very much that the idea that mathematics can explain nature, but nature also behaves according to the rules of mathematics. In other words, mathematics are sort of like this ontological reality, and that we're discovering the structure of nature when we use mathematics. And so that's kind of the point of view here, where people are coming from with respect to cellular automata. But this is a nice simulation. This is a grid size of a 208 by 208 grid, so that's a lot more cells. And of course, we still see patterns emerging, but we don't see things that are like greatly different. We have 5 different species that are different colors, and you have the model depth, the model width, the cell state dimensions, the kernel size. We can use different optimizers. We can use SGD, atom, RMS prop, SGD plus momentum. So I don't know what those are. You'd have to go look in their documentation for that. Now, this resembles another platform, which I've talked about in other meetings, which is the et.al platform. And this is where they're more directly modeling artificial life, but in which this grid exists, and each cell of the grid is an organism or a digital organism with a genome inside. So it's not a set of rules. It's not a convolutional neural network. It's actually a model of a circular genome or something that you'd find in bacteria, where you have this encoding that has a number of genes, and those genes encode different properties of the organism. And so you can achieve things that are very similar to this in that kind of an environment. And in et.al, it isn't really a cellular automaton. It's actually just a bunch of these organisms interacting in a space on a two-dimensional space. It's toroidal, but so it's similar to this. But it does actually produce, like visually, if you're just looking at the patterns it's producing, it produces very similar results. So this is considered artificial life. This is the Petri dish NCA. This is Sakana AI. This is one of the things that they've been working on.